Okay, good morning, everybody. It's a pleasure to be here and present Alexander Purcell, who will give us a seminar today. He's best called Sandy Purcell, and we are very happy to have him here with us. He will talk today about his passion. His passion is Xylella fastidiosa, you know. He has been working with this bacteria for many years, and also with the insect vectors that transmit the bacteria. He got his BS from the Air Force Academy, and after 6 years as a pilot, he got his PhD in entomology from the University of California at Davis in 1974, where his research was on Pierce disease in Napa Valley. Then he became an assistant professor at UC Berkeley when he worked on Xylella diseases, also phytoplasma diseases, and finally aphid secondary syndrome. Sandy is an emeritus professor now, working at Berkeley, and collaborates with his former student, Rodrigo Almeida, that you know him very well. Sandy now says he's Rodrigo's student, but he's still working in Xylella in California. He's well-known Xylella expert, and he's traveling still to many different places. And we have the pleasure now to have him in my lab for some days, and he will have the opportunity to present his seminar today. Thank you very much.
Okay, thank you for that introduction. Can everyone hear me okay? Yeah. And I'm really happy to be here. Thanks for the people here at CSIC in Madrid for inviting me. And I've had a wonderful time so far, and I've learned a lot here. Okay, so today I will talk about some aspects of Xylella that are already known. Many of you will not learn a whole lot new, but I hope everybody learns a little something new that we have. What I'm trying to do today is to take a different look at the epidemiology of Pierce disease from the particular viewpoint of water and temperature. So you see these first things here. You've got the ocean water, no plants, no insects. Desert, no plants, no insects. So it's obvious plants need water to grow, and insects that we're involved with today, talking about, need certain plants as well. So clearly, water and climate are essential parts of all plant life and insect life. But we're going to look at available water, because even in the desert, you have water that's available. Here is in Southern California in the Mojave Desert, a plant, the Joshua tree, that supports a particular insect. Even without only 17 centimeters of rain a year, there's an insect that lives on the Joshua tree. The Joshua tree sharpshooter lives only on this plant. And somehow it gets enough water to support itself. And it's eating 200 or 300 times its weight every day in water, maybe more sometimes. So water is very important to that insect. Even in the desert, you have very often enough water for this disease to exist. It's where the water is available. Well, this is a very traditional look at plant diseases. It emphasizes that the presence of a pathogen in a plant does not necessarily mean disease. Xylella is a wonderful example of that. In fact, it usually does not cause a disease. Only under unusual associations, new associations, do you get a disease. We'll talk about that. But the environment is very important. In this case, it clearly determines whether or not you will have a disease. If we introduce a vector into the equation, this changes things. It makes it more complicated. Now, instead of 4 different interactions, you have many more, at least 6 or more. So we're going to concentrate on just two environmental variables. There's variables in the pathogen, there's variables in the vector. We're going to look at that with respect to water and temperature. First, water in the soil, in the plant, and in the insect too. And temperature will relate to all of these as well. First, let's look at Xylella, the bacterium, and the water environment. What is it about this environment that's sort of special? Maybe not unique, but special for Xylella. First is the water in which it lives is very dilute chemically. It's mostly water, over 99.9%. So it's less than 1% of that's not water. Therefore, it experiences a high osmotic pressure. Most wall bacteria are able to deal with this. The osmotic pressure attracts water into the cell. In this case, the bacteria has a way to deal with that, a rigid cell wall. It also has a dilute nutrient environment and it has to have special ways to deal with that. And it does. It does not able to move in water alone. It has no, excuse me, has no flagellum or flagella or cilia. So it just floats around in the water. They like to stick to each other, the bacteria cells. But on a solid surface, it's able to move quite well. It's what's called twitch mortality. And one end of the bacteria has these various attachment methods. It is able to attach very firmly to the insect. It attaches an area where the bacteria attaches to the mouth part of the insect, and that's the narrowest part of the food canal. It's only a few microns across. And at the center of that flow, it's as high as 5 meters a second. An insect that's 5 to 7 millimeters long is supporting velocities of 5 meters a second at the center of the food canal. Now, and theoretically, on the wall of the food canal, velocity is 0. Well, that's where the insect's attached. But it doesn't attach laying down on the cuticle. It stands up straight on end. It's maximally exposed. It's covered with a gel that probably protects it to some extent and may be involved in absorbing nutrients from the environment. In fact, it does best in the fastest part of flow. Where this flow is slow, it doesn't do so well. Very interesting, curious method. There are many genes, and now it's easy in culture to knock out genes for various genes, individual genes. And in this way, you can see which ones are essential. And turns out a number of different genes are responsible for attachment and for movement. Xylem sap is probably the simplest chemical fluid in plants. It's much simpler than others. And this may be a reason why it's able to survive and even multiply in many, many plants. In fact, most plants. If you take a needle and punch through a little drop of suspended bacteria, you can usually infect 80 to 90% of the plants you test. This will vary with the genotype. For example, there are genotypes of Xylella fastidiosa that will infect citrus, but they infect grape but don't cause disease. Others will infect grape but don't infect citrus. So it all depends. We have these long host plant lists like EFSA, but it doesn't show the important features, which is the genotype is one of the most important features of what's on a host plant list. So this feature was first discovered in the 1940s by Freytag at Berkeley. And they found using insects alone and thinking this was a virus disease, they could feed infected plants of infected vectors on a plant, take them off, put non-transmitting vectors which they tested beforehand to make sure they were infected. And if they prove infected, they throw that result out. But in this case, they found by insect transmission alone, most of the plants they tested had the virus. That's about all they knew about it. But they also found vectors collected in nature far from agriculture. Some of them had this virus as they thought it was. Now, among these 70 or 90% or high percent of plants that are propagative hosts, that is, you can show the bacteria multiplies when you introduce in the plant. But does it move in the plant? That is it systemic? Well, it turns out maybe 50% of those will be systemic hosts. And of the systemic host, only very few will cause allow the bacteria to move systemically in the plant. Or even rarer is cause harm, disease to the plant. Most of these plant hosts have no symptoms, at least visible symptoms. And from a lot of these hosts that prove to be systemic, propagative, and systemic, eventually the bacteria disappears from the plants. Some plants, it stays on and on for years. So here's an example. This is an old slide I've used from a study by Tina Wistrom and myself some years ago. We tested 29 common weeds found in vineyards in Central California, vineyards or near them. So grape, shown here on the left, in yellow, 100% infection, 100% systemic movement. So the systemic movement's in yellow. And of these, I've arranged them in order of percent infection and systemic movement. And so what you get here is, for example, horseweed. It's in the sunflower family, the Asteraceae. Sunflower is an excellent host, and it's one that's very persistent host. But some of them recover. That's not shown here. Here's an example of a systemic host, tomato. You look, and sure enough, the bacteria, when you see this many bacteria in a cell, in a plant, this many Xylella bacterium, those bacteria are all dead now. You know, you stain it with once it reaches that level, they die quickly. They're too crowded. So they have to move. And here is a engineered Xylella to produce fluorescent green cells, so we can easily see them under fluorescence. And here are the pits from one xylem cell to another. And you see every pit is occupied by Xylella. So they have a special way. It's a narrow fit. They're barely fit through there, but they do. They have to, there's a membrane at the pit keeping air from moving from here to here. If this should be filled with air, there's a pit will stop that air from moving and destroying the movement of water. In this case, it's able to get through that pit membrane. And look, it's very efficient at doing that. That's how it moves in the plant. With respect to water and vectors, xylem sap is their main food. It has very little nutritional value per milliliter. So they have to eat a lot. They eat more than any terrestrial animal, probably. The Graphocephala leafhopper has been recorded to have 1,000 times its weight a day in food maximum. The glassy wing sharpshooter, Homolodisca, it's only about 300 times. So imagine your weight, 300 times your weight, and you drank that every day. That's a lot. So what is in there? What is it trying to grow on, live on? Mostly amino acids and simple organic acids. There's some other mineral nutrients, of course. That's the fertilizer for the plant, so to speak. And the digestion of the amino acids is amazing. You measure what goes in and what goes out. You measure a certain amount, let's say, of glutamine, one very common amino acid in plant xylem, and one that the insect really cues on. It loves glutamine. It can taste it. And there's no detectable glutamine left. We don't, what the detection limit's 1 or 2%, whatever that is, it gets at least that much, 99% usually. And so they are very particular about where they feed. They chemically detect immediately at the front of their mouth. They have taste receptors, and they can either continue feeding or go to something else. And they like succulent tissue. By succulent, we mean it usually means growing tissues. And these are nutrient sinks, so they tend to have higher concentrations than more mature tissues, like older leaves. And so the nymphs, as the stem lengthens, for example, in a grapevine, they will move along with that growing tip, and they prefer those young leaves. So that's a very important feature. And to have that succulent tissue in California, for example, and places here in Spain and all over the Mediterranean, you have a climate with no rain or very little rain in the summer. And so you need soil moisture to sustain that succulent growth. And we see the insects will do that. I look at weeds along a river in the summer in California, and the particular sharpshooter there, the Brachycephala, be all over those weeds. You go into the vineyard or a field 50 meters away where it's not near the soil water in the river. There's no sharpshooters there. They have to have that succulent growth. Very important. And so the water availability is important for that succulent plant growth to be attractive to the vectors. The xylem feeding specialist, and I say xylem feeding specialist because all leafhoppers feed on some xylem at some time in their feeding. And the xylem feeding specialists are no exceptions. The Brachycephala is a vector of a phytoplasma disease, X disease. So that's proof that it does feed in phloem sometimes. But if you look at its excrement, it's all water, or almost all water. So that's mainly what they do. That's why I say a xylem sap feeding specialist. They're not completely exclusive. On the other hand, aside from being a wide spectrum or general feeder in plant selection to feed on, this insect right here, the Joshua tree sharpshooter, it lives only on Joshua tree. So it's an extreme specialist. So you have both kinds, but most xylem sap feeders specialists are generalist. Now, vectors in water. Again, soil moisture, thus I've explained, I think, how the soil moisture content is really important in attracting vectors to plants. And the manipulation of soil moisture we call irrigation. And that's a really important feature in where Xylella diseases occur, particularly in dry climates where there's a lot of irrigation and very little natural rainfall. Let's look first at the natural soil moisture. What does that do for vector populations? Well, it is simple. If you have no water or not enough water, you're not going to have the vectors there. It's that simple. They can't, in fact, they just can't survive on plants that don't have enough water. Costa Rica in Central America, they got a lot of water, and they have a great diversity of plants and vectors. They easily have more than 100 times the species of xylem feeding specialists in Costa Rica alone than the whole rest of North America combined. Canada, northern Mexico, and the United States. And the plants on Costa Rica is, you appreciate, especially those who know the early Spanish explorers. This is the El Dorado of Xylella in the world, probably, is Central America. In Florida, here's an experimental vineyard in Florida. Every plant's a hybrid trying to come up with resistant varieties. One of their most successful resistant varieties is this one in the forefront. You see it still has pretty bad disease, but it's alive. Everything else is dead or is a new plant. Plenty of rainfall, plenty of vectors, and no plants, European grapevines, can survive in this climate for more than a year or two, period. It's 100% disease within 1 or 2 years. So that's how important the natural rainfall is. No chance of raising grapevines in this environment unless you have resistant varieties, which we now do, fortunately. So in dry summers, you depend on rivers and springs to provide the moisture you need to have the disease. And Napa Valley is a good example of this. So here's an aerial picture of a vineyard in Napa Valley. Here's the Napa River. And the vector, the Graphocephala punctata, we call it the blue-green sharpshooter, common name. It moves in and has this pattern. Every vine space here has either a dead plant, no plant, a new plant, and but you look here away from the water, there's a pattern. This is 20 years of Pierce's disease, and yet only here and there you lose a few vines to Pierce's disease. Some vineyards out here, I've seen situations where the vines are 100 years old. So this pattern has been around for a long time. What causes that pattern? That's what I'm interested in. Look at Puglia, where we're going to take a look now at, this is a drone taking a video of an area in Salento in the heel of Italy. And you can see these vines, as far as you can see, are dead. Well, they look dead. They're actually still alive. They have, from the roots, are coming shoots, but eventually those too will die. Okay, so that's pretty impressive. That's the most impressive epidemic plant disease I ever saw. Here we think is what the sequence is. It's the current hypothesis. In the winter and spring weeds in this area, you find the spittlebug, Phalanus spumarius. It's common all over Europe and occurs from Finland to North Africa and into Asia. So it's clearly the most important vector of this bacteria to olive in southern Italy, in Puglia. The eggs are laid by the adults in September, October, November. They overwinter. As soon as the rains come in the winter, this Mediterranean climate, you get the weed growth that supports the growth of the eggs hatch. They become adults into early April, into maybe hatch into May. I don't know how late the latest ones do, but eventually without rain, as the rain becomes less in the spring, late spring, and then the summer, the weeds dry up. And why is that? They're running out of soil moisture in the first 50 centimeters of soil, perhaps. And so that's not attractive to the spittlebugs. What do? They go to trees, and they are tapping a much deeper level of water. And so the insects go there to feed. Eventually, they don't have enough water either. And what do? They go to other trees that seem to provide more moisture. That's our theory anyway. It's clear that's what they do. If that's why they do it, we're not completely sure, but that's the hypothesis. So eventually the trees shut down. That is, basically they reduce drastically their transpiration. And so the insects fly to other trees. And the cycle repeats itself. They will return, or they will enter the orchards and lay eggs for the next generation. So one generation a year. So you can see that the lack of rain, and of course this driven also by high temperatures, can slow or prevent the spread of disease, or they can promote it. So the change of the availability of water to the plants, as we saw from the weeds to the trees, that's That could be important. But what can change that? Irrigation. So where the natural rainfall does not supply the necessary ingredients for Xylella to spread and move around in different plants, we change that. It's called irrigation. And here's an example. This is in Baja California, Mexico. Guadalupe Valley has a very dry summer, and but there's agriculture because of the Guadalupe River here. Let's see, let me show it here. Okay, and the river only has water above the surface. Some years never. Other years maybe for 2 months, 3 months. The rest of the year you can drive a truck across the river. It's dry on top, but it's very sandy. Here is one part of the Guadalupe Valley where you see this leaking pipe. It produces water and grasses. And so this area here is very productive for producing a particular xylem feeding specialist, sharpshooter leafhopper, and other insects too, of course. And here's an example, not far away. You have these very sandy soils, and there is a grass. In Spanish, you would call it grama. We call it the Cynodon dactylon, or Bermuda grass in the United States. And it produces a particular insect, Xiphon fulgida, which loves Bermuda grass. The Bermuda grass probably is from Africa. The Xiphon is a native California insect, but the Xiphon loves Bermuda grass. You would think it's a natural host. It reaches, because the soil is sandy, they're raising these vines very next to the river. The soil is very sandy, so they have to irrigate every 2 or 3 days in the summer. This produces very high populations. With a sweep net, maybe 20 with one sweep. So you can imagine with these populations, you go from the grass up to the vine and back to both acquire and transmit with so many thousands and thousands of vectors in the vineyard all the time, you're going to get Pierce disease. It's going to spread from vine to vine. And that appears to be what happens. You go 100 meters away from the river, this situation doesn't happen. The same grass is not in a succulent condition, and it's not very dense, as you see here. So the solution is get rid of the grass. That turns out to be the solution. There's also another case where irrigation is a good example, and that's in Southern California, a place called Temecula Valley, where you have a lot of citrus and a lot of grape, and the two grow next to each other in most of the valley in different farms. So this is irrigated. This is irrigated. This is now the dormant season, but in the winter, there's a lot of the sharpshooter, the glassy wing sharpshooter, GWSS, homolodisca. Then as the grapes begin to grow in the spring, the insect moves from here to here. The Pierce's disease bacteria genotype does not infect citrus. Thank goodness. It's the one of the 10%, 20% that don't support multiplication even. But obviously the disease goes from plant to plant. So you get exponential spread of the disease. In 3 years, this vineyard's gone, just 3 years. One example, the first year they see 10 diseased vines, they pull them out. Next year, 100. The third year, they're stopped counting at 10,000. So it moves very fast in these conditions. The solution was to treat this with insecticide. All of the growers came together. The government supplied the money for the insecticide applications, and it saved the industry, the grape industry there. They still get losses that are pretty high, but they can live with it until resistance to the resistance occurs. Now it's 1,000 times resistant. So they have another chemical. You can predict that too will lose effectiveness in just a few years. So that's another example of creating vectors, and that applies to what I'm for this visit for, and that is an insect, Draclocephala, a new species from eastern United States that apparently has now infested northern northeast Spain and southern France. And here is an example of the damage it can cause. Here is actually was at the time I took this picture, it was in cotton, but it was alfalfa for many years. And curiously, I won't go in the details of why, but curiously, this disease in the Central Valley in this kind of situation where the sharpshooters grow in crops next to the vineyards, you see it's very bad here and it gets less and less. And this is, at least 10 years of disease. It's what I call a picture of, or the fossil history of the disease. Oops. Okay, so here is a newly planted plot. So you see that's why we don't have any disease showing. Here's another example. In the same region, you have this irrigation that comes out of what we call a standpipe, and you get weeds all around that irrigation water when it overflows, the pressure's too much. And so here's an example where that happens. You find next to each standpipe, you'll find concentrations of disease. So the source of the disease is outside the vineyard. And the insect flies just after sunset. 20 minutes after official sunset, it reaches its peak of flight. And that's when it lands on grape. It doesn't like grape. In my whole life, I've never seen one of these Brachycephalo on a grapevine, except the ones I put on the grapevine in the laboratory. And they're not great vectors to grape. They're pretty good vectors to alfalfa and other plants. So conclusion, you can see many ways in which water is really instrumental in localized occurrence of this Purcell's disease. Now let's take a briefer look at temperature. I'm trying to leave a lot of time here for questions and discussion. And temperature is quite important, and we really haven't even thought of all the ways in which it's important. Now, this is what's called the Arrhenius plant. This is created by my former graduate student, Eileen Field. And I want to show you the range of growth. So here's the range of the high end on the left and the low end on the right. And Xanthomonas is the nearest relative of Xylella. Xylella is about a 60% version of Xanthomonas. It's lost about 40% of its genes from its parent or ancestor, probably because it's got a parasitic way of life on both plants and insect. That's how it survives. Here in red, it's shown for Xylella, and in yellow for Xanthomonas. So you see that the max temperature here is about 34 degrees. Growth will not occur at 34, and the higher in temperature, the faster the rate of death. Hot water treatment, you can cure the dormant cutting, completely eliminate the bacteria with 30 minutes at, I think, 45, maybe 20 minutes, 45. I can't remember the details, something of that order. And so it's quite sensitive to hot temperatures. How can it survive in desert areas where it's a 40 degrees every day for days and days and days? But it does, and it sometimes does very well. So there's a question for you to answer. We don't know the answer to that. Low temperatures are extremely important in this disease. How is that? Well, long ago, I did a survey of mailers, questionnaires I sent to everybody in the Eastern Coast that I thought might have some knowledge of Purcell's disease. Most people didn't. This was in the 1980s, actually the 1970s. And so at that time, grapes were not very widely grown in the United States. There's not a lot of acres, but almost all these states now here in the eastern United States have some kind of grapevines, except the extreme north, like Minnesota, North Dakota, Montana. All the rest of them have grapevines of some kind, and but not here. No grapevines here. in these states, like this is North Carolina, South Carolina, Virginia, we see less disease as we go up to this area here has mountains. And as you go up the mountains in altitude, you get less Purcell's disease. The higher you go from, this isn't showing up, is it? I don't know why I don't have the. No, it's showing up on my screen, but any case, generally the Purcell's disease. Remember, this is grapevines, not other plants. The Purcell's disease generally becomes less the higher latitude you go, the higher altitude you go, and the further distance from large bodies of waters like the ocean that you move. And this is particularly well shown in Texas, but I won't go into that. This particular map was created for a talk that I gave back in the 1980s. I never published it. In effect, it's kind of a model, just a visual model as a map. But a colleague of mine, George Oster, who is a very widely known modeler, says, in effect, I can't remember his exact words, but he said, no model is really correct, but some are very useful. And I would say this is in this category. It's not precise, it's not completely accurate, but it's very useful. What if we take the same criteria, that is, in this case, the average minimum January temperature, and we plot straight lines where that occurs. So those are isotherm lines for that measure. And we apply it to Europe. This is what it looks like. Now, I must say that this map is different. And the difference is I would have to move all of those lines north, at least 150 miles, so 200 or 300 kilometers, at least, maybe more. So climate change has had a real impact on Purcell's disease already. You can see it, especially in California where I work and I'm familiar. We see that the disease has moved in its severity and intensity north along the coast about 50 miles. So what happened here? The red area is, again, corresponds to the same temperature criteria as I had in the previous map of North America. And the first place it occurred in Italy. Here it's back. Okay. So this is the first place we noticed it in Italy or in Europe. It's been here before. It's been here more than 30 years somewhere in Europe, for sure. It was in Kosovo. I talked with a guy that published that 98 paper, was it? Finding it in Kosovo. Yeah, it occurred, and it probably still occurs there. It was imported on, my story is that you never see published, but you hear from independent sources. The Serbians, Bulgarians imported California table grape varieties, and they had to import some Florida varieties, and those have no symptoms or little symptoms, but they're full of bacteria, very high populations. They're not resistant, but they're very tolerant. So where did it appear next? Oh, it was in Corsica. Now, at this time, the people in Italy told me, wow, you're a genius, Sandy. Look, the disease stopped in olives right there. Well, it didn't stop. It's going to go. It's yellow, which means moderate, and moderate could include some really bad losses on individual situations. But what was surprising to me was Corsica. You see the red and yellow here sections? The occurrence of them that they've mapped by sampling plants all over the island. And the disease distribution is just like that. Yes, they have it in the yellow area, but not very much. It's mostly in the red area. At the same time they found it in Corsica, they also found it in France. And so it's undoubtedly elsewhere in other plants. Depends on the genotype of the bacteria. It's probably in a lot of plants they just don't see symptoms in. Then Majorca. The first time I walked into a almond orchard there in 2017, I think. I saw that and I said, oh, this has been here 20 years or more, probably more. So it's been around a while in these places, but look where we first noticed it. And then in the Valenciana area, and then it's kind of widespread, and I understand, and from what I've read, in Portugal. So you see a pattern here? Yeah, every time we first discover it is in this area, but it's also in the yellow area. And there's some discoveries have been made in the yellow area now. Especially the latest is Pierce disease discovered here. A talk was given on that a few weeks ago. And it shows kind of sobering how fast it spread in from one little spot. A brief mention of this, which is very old work that was done again by my former student, Helene Field. And what we see here is the day of the year, starting with January 1st. So this represents April and May, June, etc. And we've got the percent recovery. So here you have no recovery. Here you have a lot of recovery. And you can see that here you have very little recovery if it was inoculated in April. If it was inoculated in May, yeah, you got more recovery. And then up here after that, no recovery except for these 3 points here, which were in Parlier, or the Central Valley, where it has much milder winters. So this fits very well with their idea that the severity of cold in the winter is somehow related to the rate of recovery. That means, and also I should add, that if we've made measurements of when we can detect, when can we culture the bacteria, and how does that correspond with the population in the grapevine? It turns out we really can't consistently culture it. And if we do culture it, it's very small populations until well into the month of June. So only in the month of June, you have very few adults. They mostly have laid their eggs and died. What you have is nymphs and a few young adults. So they're only able to pick up the bacteria in late June, sometimes middle of June, depends on the summer, or July. July, they can all, it's all over. It's a lot high populations, but you're not going to have symptoms for another 6 weeks of hot weather in Napa Valley. If you're in Texas, you're going to have good symptoms in June and July. So you see, that's an example of how temperature affects the growth. So what does that mean? It means that the vine-to-vine movement of the bacteria really can't occur until the insects can pick it up. And that's the new adults, and they go all over the vineyard. But the disease in Napa Valley doesn't go all over the vineyard, just a little percent here and there. It's the early inoculations that are important. So the date of inoculation is very important in grape in Napa Valley, because the vines inoculated after May mostly recover. If you go to Florida, there's very little, if any, recovery. So there the disease spreads like an epidemic. It's exponential. I point out that my colleague Kirkpatrick with his student Cal and others found that you can infect almond at any time of the year in the summer, and some of it will survive the winter and some of it won't. It actually has a little higher rate of recovery generally than grapes. The bacteria in the Xylella in almond move very slowly compared to grape, and also have a higher recovery rate generally. So low temperatures during grapevine dormancy can eliminate Pierce disease. They affect how fast you get symptoms. It depends on how warm it is. For example, in Texas, where they grow grapes, it's very hot day and night. In Napa Valley, the temperature, and even in the Central Valley, it becomes quite cool at night. In Texas, it becomes dark. The temperatures stay high. They have more degree days of high temperature, and they harvest varieties like Cabernet Sauvignon in July. We harvest in October. So that's the temperature effect that you see in grapevines. Just one example. We see the same thing in California. We see symptoms of Pierce disease sooner in the hotter areas than we do in the cooler areas. So temperature is a major driver. I just mentioned also, I notice where we have high vector populations, but it never gets very warm. You also probably, and we haven't proven this, but probably it's sometimes too cool for the growth of Xylella to sustain an epidemiological presence. In other words, the bacteria has to live, has to go from plant to plant and insect to insect at some point. And if it's too cold for it to grow fast enough to do that in a predictable fashion, then you will probably not have any Pierce disease. So I've noticed that where my daughter lives in the northern part of California near the coast. And I noticed, gee, we have an awful lot of this Graphocephalovector, but we never see Pierce disease. So you go interior just a little bit where it's warmer in the winter and a little colder in the winter, but much warmer in the summer. And you see Pierce disease, not very much, but we now see it. So I leave you with some questions, and that is, how can we best manage the water or use our understanding of its relations to manage Pierce's diseases, or Xylella diseases, I should say? And can we use it as a prediction tool? Another different thing is, how does sub-freezing temperatures cure grapevines? Who would not want to know how to manipulate a vine in such a way it completely cures itself of Xylella? Wouldn't you like to know that? I would. I don't know anything about that. I don't have any ideas. Maybe some plant physiologists can jump in and help us out. And how can we better integrate temperatures considerations to improve models to forecast disease or give us warning about when they occur? So with that, I'd be happy to answer any questions or welcome comments.
Thanks, Sandy, for the wonderful presentation. So now we'll probably have some questions. You can use the chat, the people that are listening online. And for the audience that we have here, if you have any questions, we will begin with those ones. Any questions?
How about the chat?
I can ask you a question here. Regarding the epidemiology, the dispersal of Xylella in grapevines, when you had this slide that shows how the non-colonizers, because those Draucocephala insects cannot colonize grapevines, but they can probably visit the plants. And in the way they visit, even if they don't like the plants, they can go to the xylem, feed for a few minutes, and probably acquire and transmit the bacteria. So even if you don't see the vectors, the vectors are moving from plant to plant and spreading the disease. That's the hypothesis, no?
Well, I think they come in California, in our situation, we notice it the most in alfalfa fields. Mm-hmm. And particularly when it's the weeds, and particularly grass weeds, that are attractive to Draclocephala and also Siphon. And they reproduce on the grasses, but they feed on alfalfa too. And alfalfa is a host and of Xylella. And Draclocephala does feed on alfalfa, and it's pretty efficient vector on alfalfa, not to grape. So when they are flying around right after sunset, a very light wind carries them in the Central Valley, usually and very consistently from west to east. And that's when they land on the plant. Their infectious ones will take a bite. They're not very efficient transmitting but you get thousands and thousands of these inoculations. Some of them are going to establish.
Okay, so the primary source would be the alfalfa, probably?
Yes, or other weeds.
Okay.
Yeah.
Thank you very much. So what about the chat or anybody else here? Yeah, you can read the questions.
So the first question is from Giuseppe Payes. and it says low temperature during grapevine dormancy can eliminate Pierce disease. So the question is, will this concept extend to other species or even the evergreen plant species?
Does the cold cure other plants? Is that what you're saying, basically?
Can you say that again? Can you repeat the question? Sorry. Okay, so low temperature during grapevine dormancy can eliminate Pierce disease. So the question is, could this concept extend to other species or even the evergreen plant species?
Yes. For example, almond. Almond also recovers. But we don't know. I mean, very few of these have been tested. Not many people have studied this. A lot of modelers have used the available data on disease distribution and so forth, but to come up with predictions of where it might occur. But as far as other plants, I don't know. We don't know any examples of that. Probably not in annual plants, but possibly grasses. Some grasses are host of the Xylella. So that's a good thing to point out that people could test. We don't know about the recovery. Well, we know about grape and almond. That's about it. We do know that, like sunflower, does that recover? It doesn't seem to, but most of the plants eventually do seem to recover, even annual plants.
Okay, thank you. So the next question is from Miguel Roman and is, what about the level of water for the development of symptoms in plant, at least in almond trees? A higher water level in the plant is related to fewer symptoms. Is it expected that a well-irrigated orchard will have a higher number of infected plants but fewer plants with symptoms? What is your experience about, 1 second,
Okay.
about that?
First of all, water stress accelerates the growth of xylella. When they get stressed, they really start to explode. And you can do the same thing with certain plant hormones, like gibberellins and auxins. You spray them on, and boy, it's very noticeably higher rate of reproduction. I don't know about movement, probably movement too. So that makes perfect sense in terms of the insect sees, hey, the plant's starting to die. I need to go somewhere else to survive. So it's starting to multiply and move. And two, it does the reverse. It kills the plant or it makes it very sick or it makes it unattractive to the insect vectors. They do not like, in fact, they're visually repelled by leaf scorch symptoms. That's been experimentally shown by Rashad and others. In my case, also in Florida, and I've noticed this, verified this in our greenhouse, if you keep pruning and watering and fertilizing the plant, it will never get Pierce's disease. That is, it will never get symptoms. You have to stress the plant to really get symptoms. If the plant's not stressed, it's not going to multiply much. And it's only when you stress it, it really starts to show symptoms. That's an important. So again, the availability of water is very important in the induction of symptoms.
Okay. We have a couple more questions. So from Tomás Osako, is there a risk for the rest of Europe that the disease could spread, especially in forests? Apparently, depths are susceptible. Are there examples of forest damage in the United States?
Oh, I'm sorry. I'm hard of hearing, so I can't hear you really.
Forest species.
Okay. All right. Okay. Yeah. This is a thing that's been almost completely ignored. But it probably is more important. Xylella is probably more important where it occurs a lot, but people don't even know it's there. And why is that? For example, alfalfa. Alfalfa doesn't like wet soils and the humid climate of the southeast. That's what they thought. No, it's Xylella. It's stunning the plants. There are other clovers. They get infected, but they're symptomless. So their hay crops, their pasture crops are limited. They don't even think about raising grapes. They try that, in southern Louisiana, and surprise, the vines all die right away. So they don't even know about it, but that's where it is most common. Where it's most common, nobody knows about it. Yeah. Does that answer your question?
Okay, we have two more questions from Davide Martinetti. He says, thanks for the great presentation. You show the effect of water locally, proximity to river or leaking irrigation pipes, and the effect of temperature at regional or even continental level. What about the effect of water availability at these larger geographical scales?
Okay, I'm sorry. The only part of the question I got is about availability of water. What does that do to the plants?
So what about the effect of water availability at these larger geographical scales?
On vectors?
The question is that you have shown specific examples where there are leaks in the pipes.
Oh.
But what about water availability at the large scale?
Large scale irrigation. Yeah, it just makes the problem worse. So the worst situation, for example, is an irrigated pasture for cattle. And you see that there's one great example. At one time, the richest man in the world was a guy named Getty, oil wealth billionaire. And he bought a lot of farmland. He bought and planted the biggest vineyard in the state of California. And there was one property owner that would not sell land to this guy. He had 10 acres. And so Getty said, well, screw him. I'll just plant everything around him. So he did. Well, what did the guy raise? A pasture. And just west of that pasture, it destroyed tens of thousands of dollars of vines in 2 years. And that was because you had the sharpshooter and boom, you had an epidemic. Yeah. I also did some date of inoculation experiments. His manager said, of course, he didn't tell Getty, but he said, you can do anything you want here. Well, I did. And I did inoculations with vectors in little cages held together with rubber bands. Well, guess what happened to some of the rubber bands in the hot sun? They boom. So I got to see what 5 or 6 leafhoppers could do to a vineyard, and it was ugly. It was at least hundreds of vines. Yeah. So, but it made the point of you inoculate different times of the year, you get different recovery rates, even in Central California.
Okay, we have one last question from Jose Juan Cortez Plana. So he says, very interesting presentation, but the humidity factor might not play an important role because in the desert there is no Xylella fastidiosa due to the lack of humidity. In humid environments with abundant vegetation, Xylella fastidiosa thrives. So the question is, will it be the water, humidity, and temperature triad?
What do you need to do to water to?
So if it's like a triad, like, will it be the water, humidity, and temperature triad? And maybe I'm not. Do you want to read the question?
Okay. We're having trouble with the speaker's hard hearing.
Well, I will read again the question, but I don't understand it very well myself. Very interesting presentation, but the humidity factor might not play an important role because in the desert, there is no Xylella fastidiosa due to the lack of humidity.
Yeah, well, humidity, if he means soil water, I think humidity in the air is probably not a big factor. I can't think of how it would be. It might be, but the soil water is clearly very important. I wonder if that answers the question.
The question actually is, would it be the water, humidity, and temperature triad? I think he means the interactions between water, humidity, and temperature, which is important. Well,
certainly water and temperature are important, and you're going to have whatever humidity you have. It depends on a lot of things. Humidity in the air, I'm not sure how that would play. I don't know. So I can't really, I say I don't know the answer. Yeah.
Okay, we don't have any more questions on the chat. I just have a curiosity. So about the Joshua Tree sharpshooter. So you were talking about this insect. So what will be the role on spreading the disease for the?
Okay, the Joshua Tree sharpshooter, when they tested it, they got some positives. I went out with my friend Juan Lopez, and we collected. Well, it's hard to collect these guys. They got some very sharp leaves at the end, and the leaves are about I don't know, about 30 centimeters long, and you have to have a long aspirator. And these guys don't fly; they just jump from one place to another. So they're well protected from natural predators. And so we collected a bunch of them and tested them on periwinkle and grape and alfalfa, and we got no transmission. So I can't say they're naturally infective, but that's what they reported in the 1940s. And no one, I don't think, has ever seen this sharpshooter on anything. Originally, however, they tried to plant almonds in the Mojave Desert, and they irrigated with the well from groundwater. And they lost all the trees, and they didn't know the disease. They called the disease golden death because all the trees died. And the farm advisor then, 30 years later, we took him out to see almond leaf scorch. And he says, that's golden death. So just based on that kind of evidence, it probably is present somewhere. We found the Mojave Desert back in the 40s. They looked for alfalfa dwarf, and surprisingly, they grow a lot of alfalfa in the Mojave Desert. They never found alfalfa dwarf. They found, I don't think they found Purcell disease, but they never found alfalfa dwarf. We now know there's other genotypes there. So that was interesting. And we still don't know a genotype that doesn't infect alfalfa in California, but there must be one.
Thank you.
Okay, so thank you very much for the seminar, Sandy. And I think we are over now, unless there is any other questions here in the audience. I don't see any more questions. Okay, thanks.
Okay, thank you.